Good afternoon, everyone. Today I'm going to be talking to you about laboratory diagnostics for hepatitis C. I want to start with the hepatitis C genome organization. Hepatitis C is a positive sense RNA virus with a genome of approximately 9,600 nucleotides in the family of flaviviruses. HCV contains a large open reading frame flanked by the 5' and 3' short non-translated regions, critical for viral replication and translation. Translation of the open reading frame produces a polyprotein of approximately 3,000 amino acids. The polyprotein is cleaved by viral and host proteases into 3 structural proteins, core and envelope glycoproteins, which are involved in viral particle production, and 7 non-structural proteins involved in viral processing, replication, and particle assembly. Like all positive sense RNA viruses, HCV possesses an error-prone RNA polymerase. However, compared to other positive sense RNA viruses, HCV exhibits extreme genetic diversity, greatly exceeding that of HIV. The 7 genotypes are classified on the basis of phylogenetic analysis of whole genomes, with 108 subtypes recognized, defined by less than 15% genetic differentiation. Subtypes 1A, 1B, 2A, and 3 are widely distributed across the globe and account for a large proportion of HCV infections in high-income countries, up to 80% of the circulating genotypes. These are so-called epidemic subtypes. Endemic subtypes from genotype 1 and 2 are primarily found in West Africa, genotype 3 in South Asia, 4 in Central Africa and the Middle East, and is especially predominant in Egypt, genotype 5 in Southern Africa, and 6 in Southeast Asia. Genotype 7 has only been found in a few individuals from Central Africa. HCV replicates primarily, if not exclusively, in hepatocytes. The virions attach to receptors on the cell surface where interactions between the viral envelope glycoproteins and the receptor lead to endocytosis. Fusion of viral and cellular membranes cause the release of the HCV nucleocapsid into the cytoplasm. This initiates translation of genomic RNA and the generation of the polyprotein, which is cleaved into 10 mature proteins. Viral and host factors modify the endoplasmic reticulum of the hepatocyte to form a membranous replication complex, which is the major site of viral RNA amplification. Positive strand viral RNA is synthesized from a negative strand template. A portion of the synthesized RNA is packaged into nucleocapsids where they are associated with glycoproteins, followed by encapsidation. Virions follow the cellular secretory pathway, and mature virions are released from the cell via exocytosis. Throughout the world, blood transfusions from infected donors, unsafe therapeutic injection practices, and injection drug use have been the predominant risk factors for infections with HCV. In developed countries, the primary risk factor remains injection drug use. In the United States, 3.5 million individuals are infected with hepatitis C, and only about 25% of these individuals will have clinical evidence of disease, which leads to it being called which leads to silent disease progression over several decades. Persistently infected individuals are at risk for developing fibrosis, cirrhosis, and hepatocellular carcinoma, and has a 25 to 40% greater likelihood of early death. It is estimated that only 50% of those infected with HCV are aware of their infection, and over 19,600 deaths were attributed to HCV in the United States last year through the National Vital Statistics System. Studies have shown that chronic active hepatitis C developed in 26% to more than 50% of HCV-infected patients, and cirrhosis in 8 to 42%. There was an average of 3 years, and cirrhosis developed in 8 to 42% of patients at an average of 3 years after the onset of acute disease. Longer follow-up studies indicate that cirrhosis develops in 10 to 20% of persons during the first 20 years of HCV infection. Highly effective treatment regimens with direct-acting antivirals, or DAAs, targeting key proteins have reached cure rates of up to 90% even in patients with advanced liver disease. The incubation period for hepatitis C ranges from 2 to 26 weeks with an average of 7 weeks. Most HCV infections are asymptomatic, though some individuals experience malaise, nausea, abdominal pain, dark urine, and jaundice. Clinical symptoms are indistinguishable from acute hepatitis A or B without specific diagnostic tests. ALT elevations usually coincide with symptoms if present. HCV RNA and the core antigen are the first markers to appear. This is followed by the appearance of antibodies to HCV. 15 to 30% of acute cases resolve spontaneously, usually in individuals who have infections marked by jaundice and lower peak levels of viremia. Infections with genotype 3 appear more likely to resolve than genotype 1 cases. Of patients with acute HCV infection, 60 to 70% have no discernible symptoms, 20 to 30% have jaundice, and 10 to 20% have nonspecific symptoms such as loss of appetite, fatigue, and abdominal pain. Chronic infection is defined as viremia that persists for greater than 6 months. ALT levels are typically only moderately elevated after the acute phase, frequently fluctuate through the course of infection, and HCV RNA remains relatively stable. Most individuals with chronic infection have histological abnormalities. In the United States, about 40 to 60% of cases of chronic liver disease are associated with HCV infection. The progression of chronic liver disease is usually insidious. It is slow and without symptoms or physical signs in most patients during the first 2 decades after infection. Frequently, chronic hepatitis is not recognized until symptoms appear with the development of advanced liver disease. As many as 10 to the 12 infectious virions are produced in a single day. Tests for HCV infection is performed for individuals with symptoms of hepatitis or abnormal laboratory results indicative of liver disease, as well as management of patients with HCV during therapy and in the course of normal screening. As mentioned in the previous slide, HCV RNA appears approximately 1 to 2 weeks after exposure to the virus, followed closely by HCV core antigen. Antibodies to HCV typically can be detected approximately 8 to 9 weeks after exposure. This time between the ability to detect HCV RNA and antibodies is considered the window phase of the infection. Anti-HCV will indicate exposure to HCV. A person in the acute phase, chronic phase, or recovered individual will all test positive for antibodies to HCV, whereas only individuals with active infection will have HCV RNA and core antigen circulating. Anti-HCV indicates exposure to HCV, to hepatitis C, and is present throughout the acute, resolved, and chronic phases of infection. All current anti-HCV diagnostic tests detect IgG antibodies. Enzyme immunoassays, EIAs, and chemiluminescent immunoassays, CIAs, are commercially available for the detection of IgG. These assays use recombinant antigens derived from both core and non-structural regions of the HCV genome. Serum, plasma, or eluted dried blood spots may be used in the EIA and CIAs. Confirmatory immunoblot assays have traditionally been used to confirm a screening reactive antibody test. These use both recombinant antigens and synthetic peptides as antigens and are approved for use in plasma and serum samples. Previously, the Chiron-Reba was the most commonly used supplemental confirmatory assay. However, this assay has been discontinued and there are no longer any FDA-approved supplemental confirmatory assays available. The Inolea HCV score from Fujiro Bio is a CE-marked immunoblot that uses antigens from core, the E2 hypervariable region, NS3 helicase, NS4A, NS4B, and NS5A. Recombinant immunoblot assays have several disadvantages, such as being difficult to perform and having a high percentage of indeterminate results and high cost. Therefore, these anti-HCV confirmatory assays are not often used in developing countries or routine diagnostic laboratory procedures. Rapid diagnostic tests, RDTs, are also available for antibodies against HCV. RDTs represent an attractive alternative for HCV screening and diagnosis because they were validated using whole venous or capillary blood. RDTs offer the advantage of simplicity, limited need for instrumentation, minimal training, and rapid performance at room temperature. 3 different generations of anti-HCV test kits have been developed. The first EIA for the detection of anti-HCV antibodies was developed 12 years ago and had relatively poor specificity and sensitivity. The second generation of EIAs closed the window period to approximately 11 weeks by adding additional antigens, and the third generation EIAs have decreased the window period to detection of anti-HCV to 9 to 10 weeks. This is still a significant amount of time when attempting to rule out HCV transmission in needle stick or other healthcare-related exposures. Anti-HCV assays have some disadvantages, such as the high rate of false positivity in some populations, a lack of sensitivity of detection in the early window period after infection, and the inability to distinguish between acute infection, which is ongoing, active, and viremic, past infection or recovered, and persistent or chronic infections, and a possibility of false negativity with samples from immunocompromised patients who may not have an adequate antibody response. Again, the first EIA for the detection of anti-HCV antibodies was developed 12 years ago and had poor sensitivity and specificity. Seroconversions in patients with acute HCV infection was often not detected until 3 months or longer after infection. These first generation EIAs only detected antibodies against the non-structural region NS4 with the recombinant antigen C100-3. With the development of second generation tests, additional antigens from core region, NS3, and NS4 were used. The third generation EIA and anti-HCV test currently used includes an additional antigen from the NS5 region and a reconfiguration of the core and NS3 antigens. EIAs alone have low positive predictive value in low prevalence populations. The supplemental immunoblot tests for confirmation of a screening EIA-CIA used many of the same epitopes as the EIAs and CIAs, but specificity was improved with the additional epitopes from non-structural regions, and in the case of Inolea, HVR1. In the United States, the third generation EIA test is the assay most frequently used for initial HCV antibody screening. In rare circumstances, false negativity is associated with these EIAs and includes patients with major immunosuppression, such as advanced HIV infection or organ transplant patients, patients with chronic renal failure on long-term hemodialysis, and patients with acute or very early HCV infection. Again, false positive tests occur more frequently when performing testing in populations that have a very low HCV prevalence. This slide shows the currently available FDA-approved EIAs, CIAs, and microparticle enzyme immunoassays for the detection of IgG antibodies to HCV. These automated tests offer the great advantage of improved precision, reliability, technical simplicity, short turnaround time, and high-speed throughput because of the full automation. And the sensitivities and specificities of all third-generation HCV tests approved by the FDA are greater than 99% in most circumstances. The OraQuick HCV rapid antibody test was FDA approved in 2010 as a point-of-care test for use with whole blood samples obtained either by venipuncture or finger stick. The test is approved for use with whole blood serum or plasma in individuals greater than 12 years old and can be performed by technicians with little training. Results can be obtained in about 20 minutes. A patient with a reactive test should be considered to have a preliminary positive test and should undergo HCV RNA testing. In 2011, the FDA granted the Clinical Laboratory Improvement Amendments CLIA waiver for the OraQuick HCV rapid antibody test. CDC evaluations of this test determined a sensitivity and specificity of greater than 99% for serum and greater than 97% for fingerstick. As with any screening tests, the proportion of false positive rates varies depending on the prevalence of infection in the population tested. In high prevalence populations, such as injection drug users, the positive predictive value is very high. In low prevalence populations, such as blood donors, the positive predictive value is much lower. Using a supplemental immunoblot test to confirm screening reactive antibody tests, false positivity in the U.S., which has low endemicity, has been estimated to be approximately 35% in immunocompetent populations. Again, blood donors, active or retired military, and the general population. While in the immunocompromised, rates averaged approximately 15%. Recent NHANES estimates have a false positivity rate of approximately 22%. Certain ethnic populations may also exhibit higher rates of false positive anti-HCV. For instance, in certain Native African populations, higher rates of false positivity have been reported. In 2013, CDC updated its recommendations for HCV tested, which focused on identifying individuals with current HCV infection. This was because of the availability of highly efficacious antivirals, as well as the discontinuation of Reba and the availability of an FDA-approved rapid test for anti-HCV. Previous recommendations focused on confirmation of screening reactive anti-HCV results, either with a supplemental anti-HCV confirmatory assay like Reba, or by using various signal-to-cutoff ratios established for certain assays. In 2013, we published a new recommended testing sequence for diagnosing active hepatitis C infection. The new recommended sequence consists of initial testing for HCV antibody using either a rapid or conventional assay, followed by HCV RNA testing for all positive HCV antibody tests. Persons who have a negative screening HCV antibody test are not considered infected with HCV and do not need further diagnostic evaluation unless they have a known risk factor for a false negative test, such as a suspected acute HCV infection, chronic hemodialysis, or an immunocompromising condition. Individuals who have a positive HCV antibody test and a positive HCV RNA test are considered to have current active HCV infection. If an individual has a positive HCV antibody test and a negative HCV RNA assay, they are considered to have no evidence of current HCV infection. In this situation, further testing with a different HCV antibody test can usually help to differentiate past or resolved infection from a biological false positive result. Ideally, samples that test positive with a laboratory-conducted HCV antibody test would then undergo reflexive HCV RNA testing using the same patient blood sample. Some laboratories now do offer reflexive HCV RNA testing on HCV antibody positive samples. Other approaches for follow-up HCV RNA testing include collecting 2 separate venipuncture samples at the initial draw with the option of ordering the HCV RNA test if the antibody test is positive, or having the patient return for another venipuncture after receiving a positive antibody test result. From a practical standpoint, it is clearly preferable to have the laboratory reflexively perform the HCV RNA testing for a positive HCV antibody test utilizing the same blood sample. Challenges do exist in the implementation of the revised testing guidelines. Previously, qualitative HCV RNA tests were the most sensitive on the market. However, quantitative HCV RNA tests now detect down to 12 IUs per mil. Yet not all quantitative HCV RNA tests are approved for diagnostics. The HCV AmpliPrep TaqMan HCV test recently received approval for use as a diagnostic test. Prior to this, if a lab wanted to use a quantitative HCV RNA test and report this under CLIA regulations, the test had to be validated in the lab. HCV RNA tests, whether quantitative or qualitative, are significantly more expensive to perform, requires more technical staff, large analyzers, dedicated clean rooms and procedure areas, and pristine serum or plasma samples. The question of whether the same sample can be used for anti-HCV testing with reflux to NAT has been explored. At least one state now mandates that any anti-HCV positive sample automatically reflects to nucleic acid testing. CDC Division of Viral Hepatitis tested 4 different platforms to assess the ability to reflex directly from a serum or plasma sample tested on an automated platform for anti-HCV directly to an HCV RNA test. The 4 platforms assessed were the OrthoClinical Vitros ECI 3600, Siemens Advia Centaur, the Abbott Architect i2000, and the Roche Alexis. 3 systems used disposable tips on the serology assays, while the Abbott Architect uses a fixed probe system, introducing the potential for cross-contamination. HCV negative serum was alternatively placed between low, mid, and high positive RNA samples on the serology analyzers, and these same samples were then tested for HCV RNA using the Roche Cobas AmpliPrep TaqMan HCV version 2.0. None of the analyzers which used disposable tips showed any levels of cross-contamination in the negative serum samples. However, low-level contamination on the Abbott Architect, which uses a fixed probe with several washes following transfer of the serum, was found. Negative HCV RNA samples that followed high-titer RNA samples with titers greater than 6 log 6 IUs per mil showed very low levels of RNA contamination, generally less than 15 IUs per mil, but ranging from 15 IUs per mil to 100 IUs per mil. Antibody tests indicate current or past exposure to HCV, but does not differentiate between the two. HCV RNA is the true marker of active infection. Available nucleic acid tests include qualitative and quantitative tests. Qualitative had the lowest limit of quantitation at one time, but quantitative tests now have a lower limit of quantitation down to 12 IUs per mil without a diagnostic claim, or 15 IUs per mil with the diagnostic claim. Qualitative tests for blood screening are the only tests with lower limits of quantitation. All commercial FDA-approved tests are based either on real-time reverse transcriptase PCR, branch DNA, or transcription-mediated amplification techniques. The dynamic ranges are from 5 IUs per mil for blood screening, or 12 IUs per mil to 10 to the 7th IUs per mil, and the specificity is greater than 99% for all genotypes. The quantitation of HCV RNA is useful for establishing baseline viral titers if the individual is positive for monitoring therapy in the future. However, it is not required for diagnosing HCV infection. A positive genotype will also establish current HCV infection, which can be done through direct Sanger sequencing, reverse hybridization, solid phase electrochemical array, or next generation sequencing. While not approved for the use in the United States, levels of HCV core antigen have been shown to be highly correlated with levels of HCV RNA and is considered a marker of active infection. Like HCV RNA, HCV core antigen is detectable within 1 to 2 weeks after exposure to HCV. Samples appropriate for testing HCV core antigen include serum, plasma, and dried blood spots. There has been one report of an HCV core antigen test that is in development that was able to detect the antigen in urine, but no independent corroboration of this has been found. Current HCV nucleic acid tests require greater than 500 microliters of serum. All serology testing requires much lower volumes of sample input, with the commercial CE-marked test for core antigen requiring 180 microliters of serum, including dead volume. Another advantage is that the sample can be automatically reflexed from a positive anti-HCV test directly to core antigen if using the Abbott Architect platform. As mentioned, the Architect test is a chemiluminescent test. It takes approximately 36 minutes to result and has a dynamic range of 10 to 20,000 femtomoles per liter. It requires 2/3 less serum than that required for an HCV RNA test. As you can see in this graph at the bottom, we looked at, this paper looked at the correlation between HCV core antigen and HCV RNA in a patient that was infected with hepatitis C. Core antigen levels closely mimicked that of HCV RNA even through the start of treatment. CDC, Division of Viral Hepatitis, evaluated the HCV core antigen assay from Abbott. We did an evaluation of the assay using both high-risk injection drug users and plasma donors, and the correlation of HCV core antigen with HCV RNA was greater than 95%. In samples which were antibody negative but HCV RNA positive, those samples that are in the window period and were getting missed by EIAs and CIAs, we found sensitivity was 100%, meaning that HCV core antigen was detected in all samples that were antibody positive and HCV RNA, that were antibody negative and HCV RNA positive. Samples in which there were both antibody positive and RNA positive, the sensitivity fell to about 94%. However, those samples that were missed had core antigen levels that were lower than, RNA levels that were lower than 3,000 IUs per mil. Given the lower cost for serology assays, lower serum volume required, and ability to automatically reflex from the same sample after a positive antibody screen to the HCV core antigen, Europe and countries other than the United States have explored the utility of using core antigen test for blood screening as a primary screening assay or as a diagnostic test to detect current HCV infections and for monitoring antiviral therapies. In fact, as part of an elimination project in the country of Georgia, the HCV core antigen assay is being used as an alternative to nucleic acid testing to determine the effectiveness of the direct-acting antivirals on sustained virological response. Listed here are the commercially available nucleic acid tests. As previously stated, the quantitative HCV RNA assays now have a lower limit of quantitation, approximating or better than some of the qualitative HCV RNA tests, giving no advantage to performing a qualitative compared to a quantitative test. The obvious difference for some of the qualitative tests are those which are approved only for blood and organ screening, which have lower limits of quantitation below 5 IUs per mil, but these do not have a diagnostic label. There are a few genotyping assays that are available, although most are not FDA approved for use in the United States. With the advent of the near pan-genotypic DAAs, there has been less focus on the need to genotype. However, even with the pan-genotypic DAAs, there are some differences in response to therapy for certain rare genotypes. There are some manufacturers who have combined HCV viral titers with a genotyping assay in one system. However, there's little effort to gain FDA approval, again, since the DAAs are closing in on being pan-genotypic. Of note are resistant associated variants or substitutions, which can lessen the chance of achieving SVR. Sequencing technologies have greatly improved our understanding of hepatitis C virus. Initially, sequencing involved short segments of relatively conserved regions of the genome to identify the particular genotype or subgenotype through Sanger sequencing, where mixed genotypes would be identified through 2 equal or nearly equal heights of the electropherograms. Sanger sequencing includes both whole genome sequencing or amplicon sequencing. If we wanted to get an idea of minor population variants, then we could use cloning as a technique in order to generate the Sanger sequencing. Today, in order to get an idea about the minor population variants, we can use next-generation sequencing. This has simplified the process such that you create libraries of amplicons or libraries covering the entire genome. Whole genome sequencing provides variable coverage over the entire genome such that you could have 2 times coverage over certain genes and 100 times coverage over more conserved parts of the gene. Given that certain parts of the genome are more informative for constructing population-level events, such as direct transmission, we may be only interested in those regions of the genome that give us maximum variability and therefore greater probability of detecting rare quasi-species that are linked just between 2 individuals. Within our division, we use next-generation sequencing with amplicon sequencing to do just that. We amplify libraries from the hypervariable region to determine whether rare quasi-species exist in 2 or more individuals and how likely this may be by chance given the mutation rate of HCV. Next-generation or Sanger sequencing can also be used to detect recombinants where a particular variant possesses portions of different subtypes genomes in certain genes and to detect resistance-associated variants or substitutions. Additionally, rates of evolution or evolutionary studies tracking the spread and evolution within populations and over the globe can be achieved via sequencing. As I stated in the previous slide, different portions of the HCV genome have vastly different degrees of variability. More conserved regions, such as the 5' UTR, are most suitable for the detection of HCV RNA since differentiation between genotypes is minimal in this region. Areas with 20 to 30% variability are well suited for genotyping and some population-based phylogenetic analysis. Regions that have up to 40% variability, such as HVR1, are most suitable for transmission studies. In HVR1, there is less evolutionary pressure to retain conservation, and the region provides the maximum information regarding minor variants which can be associated with probable transmission events with the highest degree of certainty. In the next few slides, I'm going to walk you through a couple of outbreak investigations that our division has been involved with using both Sanger sequencing and next-generation sequencing. In these instances where we use Sanger sequencing, we ended up doing cloning in order to generate the minor variants that you will see within these phylogenetic trees. So again, the variability of the HVR1 region can be used to establish transmission links among HCV individuals. This slide shows HVR sequences from 4 patients. Although all these patients are infected with the same genotype 1A, there is a clear-cut clustering of these sequences from one another, depending on the source patient. So you don't see any intermixing of these sequences with the other patients. The power of this technology in establishing any transmission is shown within this study. The HVR1 sequences shown in green are from a surgical technician who was diverting drugs and in the process had infected more than a dozen patients. This is shown where the green sequences are intermixing with the sequences from infected patients, as shown in red here. Sequences from unrelated patients, including controls, formed separate clusters that are not associated with the surgical technician. Combined with epidemiological data, these sequencing studies confirmed without a doubt the source of HCV transmission in this particular infected patient. And this is just a blow-up showing the 18 case patients and the surgical technician sequences that are intermixed with the 18 cases. This is another investigation where an anesthesiologist reused single-dose patient vials on multiple patients in a colonoscopy clinic. HVR1 sequences among 4 patients were distinctly clustered together with sequences from patient 1.3, whose sequences are shown in red in this black shaded box. The way that the transmission occurred is going to be shown into the next slide. So this shows the mechanism through which a single dose vial can become contaminated. The first dose of propofol is drawn from the vial into a needleless syringe through a vented spike. This dose is administered through an angiocatheter to a patient with chronic hepatitis C. The reflux of minuscule amounts of blood results in contamination of the syringe with hepatitis C. This HCV-contaminated syringe with a new needle is then used to draw a second dose of medicine from the same vial of propofol. The HCV-contaminated syringe then contaminates the entire vial of propofol used for subsequent patients and is responsible for transmitting HCV to subsequent patients. And again, as shown in previous slides, the HCVR1 quasi-species from the source patients and those from the patients who received propofol from the same vial clustered together, indicating transmission of hepatitis C through this mechanism. As stated previously, there are two approaches for next-generation sequencing: whole genome sequencing and amplicon sequencing. Whole genome sequencing, again, provides variable coverage of the different genomic regions and is useful for large-scale classification of viruses, bacteria, or parasites into genotypes, subgenotypes, and serogroups. The coverage of the genome is variable depending on the ability of the primers to bind to the genomic regions of the quasi-species or variants. This leads to staggered reads across the genome, where some genetic regions may have 100 to 1,000-fold coverage, while other regions may only have 2 to 4-fold coverage. What we end up with is a consensus sequence covering the entire genome. Amplicon sequencing, on the other hand, only targets one region of the genome, typically one with a relatively high mutation rate. The result is 1,000 to 10,000 reads or more of the individual sequences available within the host. Variants with as little representation in the pool of variants as low as 1% can be detected. Therefore, it is possible to determine whether or not a transmission event has occurred with extremely high probability. Until now, for any outbreak investigation and for determination of any transmission clusters, state health labs would need to send samples from HCV-infected patients to CDC, where we would amplify and sequence the HVR1 region from these samples and use various sequence analysis programs to generate phylogenetic trees, which would require interpretation by expert bioinformaticians to determine if there was a transmission event. We have now transformed this approach into a novel technology called GHOST, for Global Hepatitis Outbreak Surveillance Tool, where laboratories who have the capacity for next-generation sequencing and use CDC-developed protocols can generate the next-generation sequences, upload the sequences into a web-based program, and automatically receive results in this graphic format showing whether or not any transmission links existed among the tested patients. Here I show how the Global Hepatitis Outbreak Surveillance Tool was used in an HIV-HCV outbreak that occurred in Indiana in 2015. The use of this tool greatly simplified from the earlier phylogenetic trees where I was showing you the identification of an HCV epidemic that was much older than the new HIV epidemic, which spurred the investigation. As seen here, as depicted in this cartoon, transmission links are represented by these circles that are connected by these lines as sort of a network of transmission events versus the phylogenetic trees that I showed you earlier. This is the entire outbreak, and what I'm showing here is that the GHOST tool identified 19 different clusters, including this large orange one that had 92 individuals associated with it. As with any virus with a great replication rate, there are instances in which resistance can be generated to the drugs that are on the market. Given that we have highly efficacious DAAs on the market now, we have found that there have been a number of drug-resistant variants or resistance-associated substitutions that have developed rendering these DAAs potentially ineffective for treatment. Shown in this slide are DAAs that are currently in use or in clinical trials. The drugs target the non-structural proteins to disrupt replication of the virus. The DAAs are vulnerable to drug resistance, and the resistant-associated variants may occur naturally prior to DAA therapy or can emerge after drug exposure. While these DAAs are highly effective and most resistance-associated variants are lost in the absence of drug pressure, compensatory mutations may reinforce the fitness of these resistance-associated variants. The clinical impact of DAA resistant-associated variants or resistant-associated substitutions on HCV treatment failure is unclear. There are no standardized methods for detecting these RAVs or RASs, and efforts to develop methods are complicated by the high degree of genetic variability and lack of pan-genotypic protocols. Furthermore, it is obvious from this slide that any sequencing method that we would use to determine the presence of these RAVs or RASs would have to cover multiple portions of the genome or requires whole genome sequencing. The interpretation of diagnostic tests for hepatitis C are given in this slide. Any positive antibody screening test will have to be accompanied by an HCV RNA test to confirm the presence of active infection. The HCV RNA test is not a confirmatory assay for antibody. Rather, it is to differentiate current from past infection. A negative anti-HCV test in a population that is not at risk is sufficient to rule out infection with HCV, for instance, in one-time screening of baby boomers or screening of pregnant women with no risk factors. A negative anti-HCV test with a positive HCV RNA test indicates that the person is actively infected with HCV. They are in the window phase or have delayed seroconversion since they have not produced antibodies against HCV. And in the case of the immunosuppressed individuals, it just may be a case of delayed seroconversion. A positive antibody test and a positive RNA test indicates either acute or chronic HCV. Finally, reactivity to anti-HCV, but HCV RNA is not detected, either indicates resolved infection, a treated infection, a period of intermittent viremia, or a false positive antibody test. In summary, the diagnostic tests for hepatitis C in the United States are limited to anti-HCV and HCV RNA. Outside of the United States, core antigen is a useful alternative to the more expensive nucleic acid testing. Antibodies to HCV are present through all phases of HCV infection, acute, chronic, and resolved. Therefore, HCV RNA is required to determine whether or not an individual has active HCV infection. Next-generation sequencing technologies are critical tools for outbreak investigations, transmission studies, and molecular surveillance of HCV, including the detection of resistance-associated substitutions or resistance-associated variants to DAA treatments. And with that, I will be happy to field any questions.
So what is the percent of specimens that are anti-HCV antibody reactive on one FDA-approved method, HCV RNA negative, and anti-HCV antibody non-reactive on a second FDA-approved method that are truly resolved HCV infections?
Yeah, so essentially this is a false positive anti-HCV test on the first screening assay.
You mentioned that the Roche CAP-CTM has the qual claim. I have also heard that the Optima HCV Quant DX on Panther has this claim.
So Optima also has a diagnostic claim.
Yes, Optima also has a diagnostic claim.
Okay. If infection resolved, antibody reactive, and RNA non-detected, does patient continue to receive medication?
If what?
It says if infection is resolved, antibody reactive and RNA non-detected, does the patient continue to receive
So
medication?
the DAAs have standards to which a standard of time to which they are applied. So if a patient does go HCV RNA negative, they will continue the treatment through the prescribed course. Again, the idea is that we want to make sure that we have truly cleared all of the infection and that we're not selecting for resistant variants. So they will continue through the course of the prescription.